The next layer of the cutaneous membrane that we're going to discuss is called the dermis. And so the dermis is just under the epidermis. And the dermis is going to be composed of connective tissue. And remember from chapter 4, majority of connective tissues will have some degree of vascularization. So they'll have blood vessels present. Whereas epithelial tissue is avascular and does not have blood vessels. So one of the big functions of the dermis is to provide a blood supply for the epidermis that lies just superficial to it, so just above it. So we'll see abundant blood vessels running through the dermis. We'll also see nervous tissue structures called sensory receptors present in the dermis. Another important function of the dermis is to anchor the epidermis in place. And because of the abundant collagen fibers present in the dermis, it really adds strength and stability and firmness to the cutaneous membrane, to the skin. And there are two distinct layers, just like we saw layers within the epidermis. We talked about those strata. There are two distinct layers composed of two different types of connective tissue found in the dermis. And so the first layer of the dermis is called, the first specific layer of the dermis is the papillary layer. And the papillary layer is going to be the thinner of the two layers. And I'm actually going to go to this picture here to kind of show you what it is that I'm talking about. So if we were to draw and see how the epidermis dips down into the dermis, and then we have connective tissue projections going up into the epidermis. And these two factors cause it to be very highly folded and invaginated. If we were to draw an imaginary dotted line where the epidermis ends right here, okay, across this, if we were to draw an imaginary dotted line, all of the connective tissue that projects up into the epidermis, that is the papillary layer. All right, and these projections are given a specific name called dermal papillae. See, each one of these is a dermal papilla. Okay, so but it's those dermal papillae are composed of loose connective tissue, and the layer is called the papillary layer. So again, the layer of the dermis that projects up into the epidermis, that is called the papillary layer. All right, if we were to draw an imaginary dotted line, all of this is papillary layer. Each one of these projections is given a specific name called a dermal papilla. It's composed, the papillary layer is composed of loose connective tissue. All right, so why loose connective tissue? So if we remember back to chapter 4, loose connective tissue will have the bulk of the extracellular matrix will be composed of ground substance. We'll still see protein fibers present, like collagen fibers, elastic fibers, but the collagen fibers aren't arranged in super thick bundles, rather more individual collagen fibers oriented in all different directions. But the key here is abundant ground substance, all right? And remember, ground substance, we'll see lots of water and proteoglycans, and I'll talk about proteoglycans in just a second, okay? But the papillary layer is loose connective tissue, and in the dermal papillae of the papillary layer, you'll see, so I want to go to this cartoon image here, again, imaginary dotted line at the dips of the epidermis, all of this connective tissue that projects up, that's called the papillary layer. Each one of these structures is called a dermal papillae. And what we find in the dermal papillae, embedded in that loose connective tissue, we see lots of capillaries. And these are the blood vessels where we have nutrient and gas exchange occur. So abundant capillaries found in that loose connective tissue. And that makes sense because, well, it's got to be highly vascularized because the cells of the epidermis that are just next to the papillary layer, that's the stratum basale cells. And remember, what are they doing? They're super metabolically active. They're undergoing rapid and lots of mitosis to generate new keratinocytes. So they require lots of oxygen and lots of nutrients because mitosis is very energy expensive. So it makes sense that the loose connective tissue of the papillary layer would have abundant blood vessels. Having loose connective tissue means more blood vessels can run through it. Having more ground substance, fewer protein fibers, means that diffusion of nutrients can happen a lot faster through that, more watery ground substance to get up to the cells of the epidermis. All right, so form function, how things are structured based on the function it has to carry out. Another structure that we see in the dermal papillae, embedded in that loose connective tissue of the papillary layer, we see this nervous tissue structure this is a sensory neuron. These are called Meissner's corpuscles. Meissner is just the guy the individual who first described these. These are light touch receptors, and so they are able to discriminate light or fine touch. And it makes sense that they would be fine touch receptors because they're located so close to the surface of the skin, right? So they're located way up here. Well, if you remember back from the epidermis, we talked about another type of light touch receptor, but it wasn't a sensory neuron itself. It was rather a modified epithelial cell. So if we go back to this image, remember here in the stratum basale, we have this special cell type called a Merkel cell, and it's a modified epithelial cell that acts as a sensory receptor. It's associated with a sensory neuron, but itself is a sensory receptor cell. It also discriminates light touch or fine touch, and that makes sense because it's located so close to the surface of the skin. So Merkel cells, as well as these Meissner corpuscles, are fine touch or light touch receptors. All right? So papillary layer, again, made up of loose connective tissue. Bulk of that extracellular matrix will be the ground substance that's going to allow for lots of oxygen and nutrients from the abundant capillaries, the abundant blood vessels, to be able to diffuse to the avascular epidermis. And then we talked about the presence of those Meissner's corpuscles, which are sensory receptors. They respond to light touch stimuli. And then, of course, just like the Merkel cells, these Meissner corpuscles will be more numerous in areas of the skin that are more sensitive to light touch, fingertips, lips, face, as well as external genitalia. So you'll find more Meissner's corpuscles located in the papillary layer of the dermis in those specific regions of the body. And then the second layer of the dermis, and it's going to be the thickest layer of the dermis, is called the reticular layer. So we go back to this picture. Remember, I drew an imaginary dotted line right here. All of the connective tissue that is shoved up here in the dermal papillae, that's the papillary layer made up of loose connective tissue. And then everything from here down until we get to the hypodermis would be dense irregular. And you can really see how the collagen fibers begin to be more densely compacted down here of the reticular layer. You really see how much more dense they are. So imaginary dotted line from here down until we see abundant adipose. So there's no, exact line of like, this is where the papillary layer ends and this is where the reticular layer ends. But you just have to go off of what other structures or tissues are present. And when you get down deeper within the dermis and then all of a sudden you see abundant adipose is an indicator that you are now in the hypodermis. So from here to here would be the reticular layer of the dermis. And the reticular layer of the dermis is composed of a different type of connective tissue, that dense, irregular connective tissue. And remember, dense, irregular connective tissue is going to be, that extracellular matrix is going to be primarily composed of thick bundles of protein fibers, right? It will still have ground substance present, but the bulk of the extracellular matrix will be thick bundles of protein fibers, with the most prominent protein fiber being collagen. So big, thick bundles of collagen oriented in all different directions. Remember, when you think collagen, you should think a rope. It resists pulling forces. So these big, thick collagen bundles oriented in all different directions really add strength and stability to the cutaneous membrane and to the dermis, and it enables it to resist pulling forces. Now, you also see elastic fibers present too. They're difficult to see on a histology slide because the collagen just really just outweighs, like you'll see way more collagen present. Elastic fibers are present, however, and they're going to be oriented in all different directions as well. And the elastic fibers that are present in the dermis allow, if the skin does get stretched, then it can be able to recoil back to its original length. So it adds elasticity to the skin, although the bulk of the protein fibers present will be collagen bundles, resist pulling forces. Now, this rich in proteoglycans. Proteoglycans, if you remember back from chapter 4, are proteins that have these large complex carbohydrate chains attached to them, and they will be they have charges associated with them, and those charges draw water into the ground substance. So you see abundant proteoglycans in the ground substance of the reticular layer as well as of the papillary layer. And having those proteoglycans present draws water into the connective tissue of the dermis, and that water that collects there keeps the skin firm and supple and hydrated, and that's really important as well. So that's, if you aren't hydrated, a lot of times you can tell it just the way that your skin looks, and it's because under normal circumstances in a well-hydrated individual, your skin will be firm and supple because of those proteoglycans that draw water into the dermis of the skin. Okay? Then let's see. So again, the thick collagen fibers are oriented in all different directions, meaning the dermis has the ability to resist pulling forces in all different directions. That makes sense because our skin wraps around our entire body and gets pulled in all different directions all of the time, just as we move throughout the day. We see another nervous tissue structure located in the reticular layer of the dermis, and these are called Pacinian corpuscles. Lamellated corpuscles, because they have lamellae or layers, kind of looks like an onion. So these layers, I like calling them Pacinian corpuscles. This is the individual who first identified them. But look, so these Pacinian corpuscles are located much deeper within the reticular layer of the dermis, whereas these Meissner's corpuscles are located in the more superficial papillary layer of the dermis. So these Pacinian corpuscles way down here are deep touch receptors, and they sense deep pressure as well. So they're a sensory receptor, they're a sensory neuron, contains sensory receptors, but they are deep touch and pressure receptors. That makes sense because they're located way down here. It's going to take a lot more pressure and depression of the skin for these to get stimulated. So these are deep touch, deep pressure receptors, okay? And they sense vibrations as well. So changes in pressure, deep pressure, and vibration. And then all throughout the reticular layer, we will also see, if we go back to this, we will see other structures present like sweat glands, sebaceous glands, hair follicles, other blood vessels, all throughout the reticular layer of the dermis. So you should know the papillary layer, what type of tissue is present and why. The reticular layer of the dermis, what type of tissue is present and why. Form, function, all of that. Okay, structure, function, relationships. Now, as far as the skin markings, we're not going to spend a lot of time on this. Just this is kind of more FYI. I may have very simple questions on this portion of the chapter, but those dermal papillae that project up into the epidermis of the skin, and it causes the epidermis to be invaginated and folded. These are called dermal ridges, and they cause the overlying epidermis to indent as well. And we call those indentions of the epidermis epidermal ridges. Now, these epidermal ridges are more prominent in thick skin. So if you look at the palms of your hands, soles of your feet, your fingertips, your toes, you'll see more prominent epidermal ridges. And those epidermal ridges are thought to give enhanced gripping capabilities to our hands and feet, right? So those little epidermal fine ridges help with a better gripping ability. Okay, and that also gives us our fingerprint. So those epidermal ridges are our fingerprint. And how fingerprints are formed, some of it is due to genetics. So, certain families will have, certain type, these loops, arches, and whorls. These are characteristic patterns. And a lot of times genetic families will have characteristic patterns, but every single individual has a unique fingerprint. And that actually, interestingly enough, has to do with, that actually comes from you present in the womb of your mother, and how every fetus touches the womb and how they interact with the environment in the womb is going to be different. So how I touched around my mother's womb and how you touched around your mother's womb is different. And while that tissue is developing, the way you moved around helps your fingerprint be unique to you, if that makes sense. So it has some genetically determined factors, but then it's also your fingerprint is very unique to you because of how you interacted in touching your mother's womb, if that makes sense. All right, so skin markings continued. This just talks about tension lines and flexure lines. So tension lines are indentations in the epidermis that form between gaps of collagen bundles. And so these tension lines indicate or help give direction to how the collagen is oriented underneath. And that has a clinical application because, so this is a picture showing you cleavage lines. So this gives an idea of how collagen bundles are, the orientation of collagen bundles within the dermis. And the clinical application has to do with cuts and incisions. If you were to have a cut that runs parallel to these tension or cleavage lines, so here these are the cleavage lines, this incision or wound runs parallel to the cleavage lines. So this incision has a better likelihood of not gaping open and healing faster. Whereas if you have an incision that's perpendicular, so the blue lines are your cleavage lines, this incision runs perpendicular to the cleavage lines. So these collagen fibers are pulling, think of it, the way these collagen bundles are oriented, pulls open and gapes this wound, making it more difficult and longer to heal. It's more difficult and it takes a lot longer for this incision to heal because it's perpendicular to the cleavage lines. So the clinical application is that surgeons will try to perform their incisions in which they will be parallel to the cleavage lines. Flexure lines are very deep creases, and you'll find these in areas that are where the skin is very close to joints. And so if you look at the palms of your hands or the backs of your fingers, like where your knuckles are, you'll see very deep cleavage lines. That has to do with the fact that the reticular layer of the dermis is tightly adhered to structures like bone underneath. And so there's much deeper crevices, those flexure lines. All right, so deep creases in areas surrounding joints. And that's where the reticular layer is more tightly anchored to deeper structures, causing those big flexure lines around joints. And this is just giving you kind of a clinical application talking about skin wrinkles. I think I've mentioned this before, but how skin wrinkles occur, it's age-related primarily. It can be some environmental if you smoke or if you expose yourself to high amounts of UV radiation, like constant tanning. You can have premature wrinkles, but mostly it's going to be age-related, and it has to do with a couple of things. So over time, your fibroblasts of your connective tissue are going to be less active. So they're not replacing collagen and elastic fibers and proteoglycans as quickly. And then if your fibroblasts aren't making enough new collagen, new elastic fibers, and new proteoglycans to help sustain your dermis, well then the collagen fibers, elastic fibers, and proteoglycans that are present over time will start to break down and not be as good at carrying out their function. And then your skin loses its strength and it can no longer really resist pulling forces. And then when your skin does get pulled on, the elastic fibers are also starting to break down and it's not able to recoil back to its original length. Therefore, once the skin gets pulled, it stays pulled and it sags, all right? The fact that proteoglycans aren't being made and produced and the old proteoglycans start to break down, the skin is no longer, the dermis is no longer able to draw water into it. So now your skin is less supple and firm. Those are all age related. And then it talks about, you can read through this if you want, I won't ask about it, but how Botox and fillers, pretty much what they're how they act in helping reduce the appearance of fine lines and wrinkles. But the best way to avoid wrinkles is avoidance of sun or intense tanning, use of sunscreens, maintaining hydration, and not smoking are the best ways to avoid skin wrinkles.